Thank you very much for giving me the opportunity to present here at this exciting round table. So I'm talking about the thalamic nuclei imaging cognitive perspective. In our research group, we focus on understanding human communication and particularly the sensory mechanisms. Human communication relies on processing of dynamic, complex audiovisual signals in interaction with cognitive processes. So in this talk, I will focus on the sensory thalamus and human communication. So why is the thalamus interesting for human communication? One of the first evidence for that is that there is a task-dependent modulation for speech recognition in auditory and visual thalamus. This means that when we listen to speech and recognize speech in an fMRI environment, in contrast to a control task, for example, recognizing the speaker identity, we have enhanced responses in the auditory sensory thalamus, the medial geniculate body, MGB. And these enhanced responses correlate with how well participants are at speech recognition. So here's the speech recognition score, here's the task-dependent modulation of the left MGB, and there's a positive correlation. So this means people who are good at speech recognition also have a high task-dependent modulation in the left MGB. We have replicated these findings in several other studies, and we have also shown that this is not only a feature of the auditory thalamus, but that it also occurs in the visual thalamus when we recognize visual speech, so when we lip read. So there's a modulation of MGB and LGN, the auditory and visual sensory thalami, in speech recognition. In addition, there is a long-standing hypothesis in developmental dyslexia that there are sensory thalamus alterations. Developmental dyslexia is a reading disorder, but most are also impaired in speech sound processing. So there are post-mortem findings that show that in the left MGB, there are alterations of large neurons in dyslexics versus controls, and that there are also alterations in the magnocellular part of the LGN, and this is based on postmortem findings in 5 subjects. But also, in vivo studies have shown that there are alterations of sensory thalami in developmental dyslexia. I would quickly brush over them. So there is less task-dependent modulation in the auditory MGB when dyslexics recognize speech in contrast to controls. There's also less structural connectivity between the left MGB and a region that codes for auditory motion in the cortex, while the primary pathway is intact. And also in the visual modality, we find reduced structural connectivity between left V5 and MT and the left LGN, and V5MT is again a region that codes for motion. And the primary pathway here in blue between LGN and V1 is intact. These studies have too low spatial resolution to dissolve subsections of the nuclei reliably. So developmental dyslexia is characterized by sensory thalamus alterations in vivo, but histology shows alterations only in parts of the thalamus, the magnocellular layers and subsections. So our next question was whether we can actually image a neuroimage LGN subsections in vivo. And this is a study that Christa did using quantitative MRI. Here's a histology section of LGN, and here in blue is the parvocellular layers of the LGN, and in red are the magnocellular layers. And what Christa did is that she used high resolution in vivo quantitative MRI to look at the T1 relaxation time to get at different myelin content. And what she found was that the LGN subdivisions can be mapped based on the differences in myelinization, that there's lower myelinization density in the magnocellular LGN, and this was validated by postmortem histology. And Christa also derive probabilistic maps of the LGN and its subsections, and they are available on NORSIF. So the next question we wanted to ask is whether the alterations that we find in this dyslexia are specific to LGN magnocellular subsections in vivo. And for this, we adapted a design by Rachel Dennison, where participants look at stimuli that are optimized for revealing M responses in the LGN as well as P responses in the LGN, also passive viewing design. And we localized the LGN using the probabilistic LGN atlas that Krista had created previously. And we then mapped the M and P maps, so the responses that are higher for magnocellular or parvocellular stimuli. And here are examples for controls and examples of dyslexics. And we wanted to know whether there are alterations in the cellular subdivision parts in dyslexics, particularly in the left LGN, in contrast to controls. And these, very briefly, the results. These are the board responses separately for the magno and parvocellular subsections in controls versus dyslexics. And for the left and the right hemisphere separately. And what one can see here is that there are differences in the left versus the right hemisphere in the magnocellular subsections in dyslexics, which is not the case in the parvocellular subsections. And this difference here is also different from the responses in the controls. So there's a group subdivision hemisphere interaction, and there's a difference between the left and right MLGN in dyslexics, but not in controls. So yes, there are alterations in LGN magnocellular subsections in dyslexia, but not in parvocellular subsections. So with this, I would like to end. Thanks to my group, the members of the Chair of Cognitive and Clinical Neuroscience, here especially Krista Müller-Ax, Begonia Diaz, and Nadia Chencha, because they have contributed to the studies that I showed in this talk, also to our collaborators and our funding. And thank you very much for your attention. And before I end, here are my open questions for the roundtable discussion. Thank you very much for listening.